I'm going to run through a kind of small subset of the work we're doing for supporting polio eradication today. So this is work with a team in the DRC, the Institut National de INRB. I never get the French right. Essentially, we've been doing poliovirus sequencing training with labs across the world for the last few years. And this is a kind of example of the implementation of this process where it's been done successfully now for a couple of years. So I'm going to run through the current state of poliovirus surveillance, how it's done and how we're trying to build on it. Looking at the direct sequencing method we've been training labs in. I'll look at the implementation of this in DRC and then how we're thinking of expanding this further to other organisms. So, why we're looking for poliovirus is to inform the current eradication effort that's been going on for the last 6 decades, maybe. It's been conducted within the Global Poliovirus Laboratory Network, so a collection of labs worldwide that tries to monitor poliovirus so that there can be vaccine responses where needed. This has been greatly successful. So in most countries, polio is eradicated. The wild-type virus is gone. So the function of these labs in many countries now is to continue to confirm that polio is eradicated so that if there is a small outbreak, it can be stamped on immediately before it has a chance to spread. The main viruses they're looking for in these scenarios is the wild-type poliovirus. We've already eradicated types 2 and 3. And also to identify strains of the vaccine virus. This is the oral poliovirus that can actually circulate in populations of low poliovirus immunity. So if it's circulating, then they actually respond with more vaccine to try and stamp it out. So this is only a problem if you've got low population immunity to poliovirus. The forms of testing, typically we have testing of stool samples, so cases with acute flaccid paralysis. Those children will provide a couple of stool samples which are tested in these labs, and also increasingly environmental surveillance, so looking at wastewater, because that's a good way of getting an idea if there's silent circulation in the population. This is the current testing algorithm. It's quite complicated. It takes a good few weeks to get through it. It involves basically trying to grow the poliovirus in a cell line that is susceptible to polio. So it's done in tissue culture flasks. You would get your stool sample in, you juice it to get the supernatant off, which may or may not have polio. You introduce this to the cell lines in several replicas, and then you wait to see if the cells die. Then you subculture that, do it again, see if they die some more. At the end of it, you know that you've probably got a poliovirus there in the sample. You don't know what type it is. It might be a vaccine strain. It might be a wild virus. So there's a series of qPCR steps to try and get an idea of the serotype, whether it's a wild virus, whether it's a worrying vaccine strain that's been circulating and unraveling, or whether it's just the vaccine strain, which on its own isn't necessarily a problem. Okay, so you've done your qPCR, maybe you're 2 or 3 weeks in at this point. To then know, is it a particularly worrying vaccine strain, you have to sequence it. Typically, this is done at a small number of global specialized laboratories where they sequence the virus using Sanger sequencing in the US or in South Africa. So if you're a lab in the DRC, you then send it abroad, and maybe 3 weeks later, you get a sequence back. So this entire process, from the stool sample being collected to actually knowing is there an outbreak you need to respond to, can take 4 to 6 weeks, which isn't great when you've got an expanding outbreak. This is all response time that you're losing. So apart from the time concerns, we're losing a lot of potential response time. You've also got a bottleneck for diversity when you're growing these viruses in these flasks, because typically one strain might outcompete the other, and you'll miss anything else that was actually in that sample. So the approach we took, this is starting about 5 or 6 years ago now, was to try and remove that cell culture step and go straight to amplifying the virus directly from stool samples or from wastewater. So we would get that supernatant, do an RNA extraction, and then use a nested PCR, like this system here, So we have a first round PCR amplifying up the capsid region. This targets enteroviruses in general. And then we have a second round PCR that is polio specific. So this is using a pair of primers used in the GPLN already, but we've tagged on some barcodes onto the edge of the primers, and these are ones used in the nanopore kits. What this means is we can then throw all the samples together in a pool and add the adapter and sequence. So it's very streamlined. We don't even bother checking at this stage after the PCR if anything's positive or negative. We just pull everything, sequence it. Minimize the chance of missing samples or of human error. Once the sequencing's done, we have a analytical pipeline called Piranha. This is generated by the Andy Rambo's team at the University of Edinburgh and has been created by OneuroTool. So this does all the heavy lifting for the labs at the end of this process. So they've got Minnow to base call demultiplex. Then the data goes straight into Piranha. All they need to provide is a barcode CSV file. So this is just a list of samples and barcodes, and point it to where the raw data is, FASTQ files. And Piranha will map all the reads to a reference database and generate consensus sequences. It's all very streamlined. It's run in a GUI, so there's not even any command line usage. So we've been doing training for labs across the kind of Eastern Mediterranean region and the African region. The training in DRC we actually did first of all during the pandemic, so we had to do it remotely. This is basically us sitting in front of a call for about a week, going through the entire process a couple of times, with the lab watching them doing it over the connection. And then fortunately we managed to follow that up in person in May of the next year. So actually going there and training them again in person, revalidating everything. Now, during this period, the team at INRB did a huge amount of work. So they were doing regular cell culture testing for poliovirus on stool samples. They did this direct sequencing method for the same sets of samples. So they did almost 2,500 stool samples over that period. 26 sequencing runs, each with about 90 to 96 barcodes all multiplexed. They just sequenced everything they got their hands on. So I've got a table here showing the cell culture results on the left. So the different Sabin strains are the vaccines, Sabin 1 and 3. VDPVs are the vaccine-derived poliovirus. So this is the one that's starting to unravel. They actually had some cases of a serotype 2 VDPV during that period in DRC. And on the x-axis are the results we got by this nanopore sequencing method. You see in bold, we've got very good agreement between the two methods. And we even picked up some extra viruses by the direct sequencing that were missed by cell culture. So excellent sensitivity and specificity. And also, we were on average getting the sequence in about 14 days. So that's from the stool arriving in the lab to getting a sequence. Far quicker than the average. This was about, 4 or 5 weeks almost by the regular algorithm at that point. So we were saving 23 days that, they could have got advance notice that there needed to be a response. And that can have a lot of implication for how many cases are generated during that time period. Now, this is probably less of concern now, but at the time, the worry was, well, you're doing nanopore sequencing. What about the error rate? So we did a comparison of their Sanger results with the nanopore results. And again, these are all sample pairs done by the two methods, and the sequences from the same sample clustered together very closely, usually within 0 or 1 base pairs over the VP1 region that we were sequencing. And just to follow on this, we didn't only just sequence the VP1 region, which is this polio-specific bit. I also mentioned the first step in the PCR is generating that capsid region. You can then just sequence that with native barcoding. And this is the huge wealth of different enteroviruses we found in a set of 8 wastewater samples. This is from a different study in Pakistan. But you can see there's a whole host of things there that are available to look into. poliovirus is only a tiny component of that, which is why we didn't go forward with this. But just by switching the primers around, these labs can actually then generate data on a whole host of pathogens. And now they're all going to be well trained in molecular sequencing and can do that kind of work in-house and generate their own data. And this is just to thank everyone involved in this process. So the team at INRB, our team at Imperial, and then the collaborators based around the world who are supporting this effort. Thank you.